Okay.
All right. Welcome to the Pacific Northwest chapter of AWG's Winter Zoom meeting. Our speaker this evening is Dr. Carolyn Stromberg, the Estella B. Leopold Professor of Biology and also the curator of paleobotany at the University of Washington's Burke Museum of Natural History and Culture, where she leads the Stromberg Lab of Paleobotany and Paleoecology. Carolyn holds degrees from Lund University in Sweden and a PhD from UC Berkeley. She has received several awards, including the University of Washington's Outstanding Undergraduate Research Mentor Award and the Charles Schuchert Award from the Paleontological Society. Tonight she'll be speaking to us about the evolution of grasses. Please join me in welcoming Carolyn Stromberg.
Thank you, Marcia. I am very excited to be here. Um, I first want to start by acknowledging that I and those of us who are in Seattle are on the traditional lands of the Coast Salish peoples whose ancestors resided here since time immemorial. Many indigenous peoples thrive in this place, alive and strong. Okay, so I'm going to be talking to you about grasses and grasslands, but first, let me just um, sort of tell you more generally what we're interested in my lab. So we're paleobotanists, and we're interested in the role that plants have played in the assembly of the ecosystems that we see around us today on Earth, and how vegetation has interacted with climate, the atmosphere, and animals to create those ecosystems over geologic timescales, of course. Um, we're talking about deep time. And we are particularly interested in a set of ecosystems in the so-called grassland biome. So this includes tropical savannas, temperate grasslands, woodlands, and so on. Um, and we're interested in that because grasslands are both unique and important. And just so you know, when I say grassland, I mean sort of grass-dominated habitats in general. Um, they are unique and important. Today, grasslands cover a large part of Earth's terrestrial surface, so over 40%, and those are all these colored fields on this map here. And as such, they provide habitat for over 1 billion human beings and many, many millions of other animals as well. And they also provide most of what we eat today, so that includes cereals and rice and corn as well, among other things. And they also have an influence on Earth's climate through their albedo and water relations, and they influence geochemical cycles, so the carbon cycle as well as the silica cycle, for example. So grasslands today are really important for us and for Earth in general. And so I've been really interested in where did these interesting and important biomes come from, or this important biome come from. So how were grassland ecosystems assembled and when, and also what were the driving factors? And this is not just something I'm curious about. I actually happen to think that it has importance for how grasslands might change in the future and how they are maintained today. So, as an example of this, when we look at how savannas, tropical savannas, are situated in climate space. So, here are two graphs with the x-axis relating to temperature and the y-axis relating to rainfall. And as you can see, savannas cover a certain part of climate space, but when we compare across continents, so these different colors here, we see that there are some differences in the distribution. So, for example, Australia here and South. I don't know if you can see my cursor. If you can't, that I will. Let's see. Maybe I can make this in.
Yes, we can see it.
You can see it. Okay, great. Then I will just keep using that. So at any rate, there is not very much overlap between South America and Australia in climate space when it comes to savannas. And those differences we think have to do with things like the bedrock, the soils, and the animals and other plants that are in those regions. So in other words, it has to do with the evolutionary history of those ecosystems. So, in other words, history matters for the legacy of those ecosystems and how they respond to climate change today and in the future. Now, for those of us interested in reconstructing the evolution of grasslands, as a paleobotanist, you look for direct evidence in the form of plant fossils, but unambiguous grass fossils are actually quite rare until about 10 million years ago, the late Miocene. So grass pollen are quite abundant, but the problem is that grass pollen look all the same within grasses, so you don't know which grass you're looking at. And some of the closest relatives of grasses also have very similar looking pollen. So they're not quite as useful as one would like. Grass leaves are almost never have the preservation, the anatomical preservation you need to really tell if it's a grass leaf or some other monocot. And then grass fruits are very diagnostic, but they are rare again until the late Miocene. So instead, what people have done is they've looked at indirect types of evidence for grasslands, and in particular, the evolution of functional morphology in large hoof mammals or ungulates, with a lot of focus on cheek teeth. So cheek tooth crown height in particular. So cheek tooth crown height ranges from low crowned, such as our own teeth, or brachydont, that's called, to high crowned or hypsodont. And there's another kind as well, but we'll focus on this for now. And so so this is a hypsodont cheek tooth from a horse, such as this zebra here, and this is what these hypsodont cheek teeth look in the jaw. And so as the animal is feeding on abrasive things, these teeth are worn down, and because they're very tall, they're kind of they last longer, so it prolongs the functional life of the tooth. And so an observation that people have made is that animals with these types of teeth are common in grasslands. And the idea is that these animals are encountering a lot of abrasive materials, both silica-rich grasses, and also when they're feeding really low to the ground, as they do in these open habitats, they get a lot of soil and dust in their diet. So based on this, the assumption has always been that hypsodonty was an adaptation to feeding on grasslands, feeding in grasslands and on grasses, because of this abrasiveness of the environment. And given that, paleontologists have used the evolution of hypsodonty in different regions on different continents as a marker for when they thought that grasslands may have evolved or spread on those continents. And as you can see, there's some variation between continents when that would have happened based on the faunal record. So what we've done in my lab is focus on using another kind of plant fossil, namely plant silica or phytoliths, so which literally means plant stone. Phytoliths are microscopic silica bodies that form in and around cells in living plants, such as this grass here. So this is the outermost layer of the grass, the epidermis, and you can see this little silica body here that when the plant dies or a leaf is dropped and it decays in the soil, these phytoliths are left after because they are more resistant chemically and mechanically than the organic material. So they can stay for hundreds to millions of years in the soil and provide a record of the plants that were living in that area. When we think of phytoliths as what the kinds of information that they give, they're sort of, the specificity taxonomically varies between taxa, and so they're not very good for giving you a list of the trees and shrubs that grew at a site. However, they have been shown to be very useful for reconstructing aspects of vegetation type. So whether you're looking at a savanna or a grassland or a forest. So they give you that kind of information. Also, within the grasses, family Poaceae, they are taxonomically quite useful. So this is a family tree or phylogeny of grasses here, and you can see that there's quite some variation here in the phytolith shape. And just to sort of walk you through the grass family, we have here at the most early diverging grasses, as well as some other grasses such as bamboos and rice and relatives, are ancestrally adapted to closed habitats, so forest floor grasses or wetland grasses. And then there are two groups or clades that independently evolve the ability to thrive in open habitats, and that's the cool temperate Poaceae, which are the grasses that dominate in cool temperate grasslands, such as here in the Pacific Northwest, where I am, and some of us are. And then we also have this PACMAD clade, which is an acronym for some of the subfamilies contained in this group. And those are the grasses that dominate in tropical savannas and grasslands. So that's sort of a general outline for the kind of variation in terms of ecology that we see within the grasses. But the nice thing then is that we can use this variation in phytolith shape across the grasses to look at diversification of ancient grass lineages, if we find them as fossils, as well as the ecology of past grass communities. Again, if we find them as fossils and can place them in these different, ecologically different groups within the grasses. So they're quite useful. Phytoliths are also useful because they can often be preserved in deposits that also preserve vertebrates, so we can get direct connection between paleobotanical, so floral and faunal information. And that's something that we don't often get with other types of plant fossils. Okay, so with using phytoliths in my lab, we've been able to tackle some questions within the assembly of the grassland biome, or relating to that topic. One is, when did the grass family first originate and diversify? When did open habitat grasses diversify and spread, and how did this relate to the evolution of grass-eating herbivores? And then, when did C4 open habitat grasses diversify and spread? And I'll define what C4 open habitat grasses are a little bit later in the talk. So I unfortunately don't have time to talk about this first topic, so we're going to focus on these latter two topics. So let's talk about open habitat grasses. So some of the work that we've done and our colleagues have done has indicated that fossil phytoliths indicate that open habitat grasses actually evolved quite early. So here are some examples of fossils that we found and have been able to place in the phylogeny, so we know what taxonomic affinity they have. And when we use these fossils to date molecular phylogeny, so we can place them in a phylogeny based on molecules. This here is a phylogeny, again, of grasses, but at this time circular. And you can see time here. It's been calibrated by placing fossils within this phylogeny. So this goes from the early Cretaceous here all the way to today. It's a little bit hard to read. I'm sorry, but that, yeah. For the geologists here, this is maybe not something you're used to looking at, but this is basically a family tree, just circular, and then time is here on as we go out from the center. And what this shows then is that open habitat grasses in the Poaceae, so these are things that are in the open, they are all red. They started diversifying potentially by the Paleocene, Eocene, somewhere in there, so quite early on. But when did these grasses become ecologically dominant? So we think that they diversified taxonomically, but when did they actually take over landscapes? So here is let's see, I have to hide these floating meeting controls because I cannot see my slides. Okay, so when people have traditionally talked about the spread of grasslands, it's often been described as occurring as a response to deteriorating Cenozoic climates. So this is what's called the Zacho's curve. Time is on the y-axis, and on the x-axis we have global temperatures reconstructed from stable oxygen isotopes or foraminiferal shells. And what that shows is, as I'm sure you're all familiar with, that since the early Eocene, we have had globally dropping temperatures, and then in continental interiors also some aridification. And so the idea is that went hand in hand with a diversification and spread, rise in abundance of grasses, and also the evolution of large ungulates that fed on grasses in tandem. So sort of a coevolutionary arms race. Okay, so this is something that I have worked to test with my lab and with colleagues. So this is a reminder to me to give a shout out to all my colleagues and former and current grad students and postdocs and undergrads that have worked with me on these questions. These are just a very small number of those people. And so I'm going to be talking very briefly about some work we've done in the past in North America, in Patagonia, and sort of Western or Eastern Mediterranean. And then I'll also talk a little bit more in detail about some more recent work in Eastern Africa. Yeah, so let's do it. But before we get into this, I want to tell you how we reconstruct vegetation type or vegetation structure using fossil phytolith assemblages. So this is showing a fossil phytolith assemblage as a pie chart. And so what we do is we reconstruct structure or openness by comparing phytoliths produced by open habitat grasses to phytoliths produced by different taxa that we typically associate with forests. So this, we call these forest indicator phytoliths. And then we also look more in detail at the grass community composition, comparing different types of grasses, closed versus open habitat grasses, so forest versus grassland grasses, in other words, Poaceae versus packmads, chloroids, and then we can, in some cases, get even more specific. And that's important because there is actually a lot of climatic variation or sort of ecological variation within the grasses. Okay, so here is just showing some pretty pictures of phytoliths from these different groups. And something to keep in mind is that generally speaking, when you are interpreting the composition of a phytolith assemblage, grasslands tend to have well over 50% open habitat grass phytoliths. Okay, so let's first look at North America, where the evolution of horses has long been used as an indicator when grasslands spread, ever since the days of Huxley and Darwin's bulldog and so on. And so this is an illustration from G.G. Simpson's book from 1951, where he's showing how grasses evolved in North America from this cat-sized, short-legged Eohippus with very brachydont teeth, so low crown teeth, all the way up to today's large, long-legged equus with very hypsodont cheek teeth. And here, what this is showing is that this went hand in hand then with these changes in vegetation from forest to grasslands. And so based on this fossil record of horses and some other ungulates as well, it was thought that grasslands spread somewhere in the late early Miocene, about 18 to 15 million years ago. So this is what we've been trying to test. And this is work I started as a grad student, and that's when I met Deb Hanneman and her husband and many other people. Okay, so what I did and what we've been doing since then also is analyze phytolith assemblages extracted from Eocene through Pliocene sediment from the US interior to try to get an idea of sort of a north-south transect through there. And oftentimes what we've looked at are vertebrate localities, so localities that also preserve fauna or that have sort of type sections, geological type sections for different formations. And that is to get good geological information from those sites. So this is diving into some data here, looking at North American grassland evolution using phytoliths. So this is a timeline here on the y-axis going from the Eocene through the Miocene. And then we have Nebraska, Wyoming here, and Colorado, so the central Great Plains. And there's some gaps here and uncertainties in the age of some of these assemblages, but more or less the pattern is relatively clear. So what we think we're seeing is that in the Eocene and early Oligocene, we have assemblages dominated by forest indicators, including grass assemblages that are dominated by what we think are bamboos. And so we reconstruct this as some kind of subtropical forest with palms and bamboos. And then sometime in the late Eocene to early Oligocene, we start seeing some typical open habitat grasses, in particular poaceae, by this time, but they are typically in relatively low abundances. So we interpret that as these grasses immigrating or perhaps evolving there, and hanging out in sort of forest glades or forest margins, something like that, but not being dominant primarily. And then sometime in the late Oligocene to early Miocene, we see these open habitat grasses becoming more dominant in assemblages, leading to some kind of grass-dominated habitats, maybe a woodland or a mosaic of a more forested and more open habitats. Because we do see some variation in the relative abundances of forest indicators versus open habitat grasses. And importantly, these earliest grasslands were dominated by cool temperate poaceae grasses, and we refer to those grasses as C3 grasses. I'll get back to that, but most grasses are of this kind, C3 grasses, but in this case, they are cool temperate poaceae grasses. Okay. We also have done work in the northern Rocky Mountains, so Montana, Idaho, more recently, and we see a similar pattern where we think that open grass-dominated habitat is starting to spread in this sort of Oligomycene time frame. There are some differences in the older assemblages with the types of grasses that are in these assemblages. We are not exactly sure where they belong in the phylogeny, but they don't seem to be typical open habitat grasses. So we see a similar shift, which indicates then that the factors that, or the processes that were responsible, that influenced this shift, may have been similar despite the difference in latitude. Okay, so if we then summarize the data we have from North America, we think that open habitat grasses diversified at least 10 million years before they became ecologically dominant in this region. And this indicates that the factors that drove the ecological expansion was not the same as what drove the taxonomic diversification. So what were the factors that drove this spread of grasslands in the Oligomycene? Well, this is a question that some collaborators of mine that we tackled by looking at some geochemistry. So this is not, I was involved in this, but I am not an expert on these methods. The lead author for this work was Tyler Kukla, who was a grad student at Stanford at the time. And basically what they did was look at stable oxygen isotopes where they were comparing clays and carbonates. And the general idea is that the oxygen isotopes vary seasonally in a predictable way. And carbonates tend to form in the dry season, in hot and dry season, whereas clays tend to reflect more the rainy season, but integrates more time. So if you compare those two, you can get an idea of when there is rainfall. And so they collected a bunch of published and new measurements of stable oxygen isotopes of clays and carbonates, and then we compared that to our phytolith data. And what we saw basically was that if we look from the west here to the east, we saw before the Oligocene-Miocene transition, they called that the open habitat transition. So this is when we saw this transition in the phytolith data that I just showed you. If we look before that time and after that time, there is a shift in the continental interior towards winter aridity. So going from here, this color corresponds to winter rains in the west coast, and then sort of gradual shifts to winter aridity. So we see this much more pronounced after this Oligomycene transition. The suggestion that we came up with that may have driven this was the idea that perhaps there was some mountain building in the Cascades. This is highly controversial, and so this was just an idea that we threw out because the timing of that is still debated, and I think most people think it happened a lot later. All right, so when we then compare our data to the evolution of Hypsodonty in North America, we see that grasslands appeared at least 4 million years before we see this shift. So we think that although it probably had an effect, it didn't have an immediate effect. So this idea that there was a sort of an arms race that was happening successively is not true. Instead, there may have been some aspects of the environment that helped buffer. So, for example, there was enough browse that the horses and other ungulates didn't have to feed on grasses initially. This is something we're still working on understanding. But at any rate, the point is that they evolved in the context of grasslands. We've also done a similar study in the Eastern Mediterranean, and there we again see this lag between the spread of grasslands locally or regionally and the evolution of Hypsodonty in that region. But the main point here is still that these hypsodont ungulates evolved in the context of grasslands. We've also done work in South America, in Patagonia, and there, interestingly enough, Hypsodonty evolved almost 20 million years earlier than everywhere else. So for a long time, it was thought that grasslands spread there before anywhere else. However, our work has indicated that there was actually, that grasses were very rare until at least some 10 million years ago. And this is supported by pollen data as well. So in this region, Hypsodonty did not evolve in the context of grassland. So very different story. And so we were very interested in what was happening, because the way we initially interpreted the phytolith assemblages was as something like a forest. So it was very strange that Hypsodonty would evolve in the context of a forest. So to test this and further research it, my then grad student, Reagan Dunn, who is now at La Brea Tar Pits and Museum down in LA, she developed a proxy for openness, so habitat openness, based on phytolith shape and size. And the way that works is that the epidermal cell, so the outermost cell on the leaves of a plant, their shape and size is influenced by light environment. So in plants that grow in full light, they tend to have smaller and less undulated epidermal cells, whereas plants growing in the understory of a forest, they tend to have very large and very undulate or sinuate epidermal cells. And because phytoliths in the epidermis tend to basically be a cast of those epidermal cells, we can use them to measure shape and size. So this is an example of phytoliths that are basically casts of epidermal cells. And so Reagan went to Costa Rica and did a modern analog study where she calibrated this method. And then we applied it to phytolith assemblages from southern South America. So this is showing, the modern study correlated with something called leaf area index, which is a measurement of openness that's commonly used in remote sensing and ecology, for example. And so she called her proxy RLAI, or reconstructed LAI, for leaf area index. And so you have that on the y-axis, and some modern ecosystem LAI ranges here, going from desert and shrubland up to broadleaf forest. And then we have time on the x-axis going forward to the right. And so what we see here is that habitats were very closed in the early Eocene, and then towards the middle and late Eocene, they became very open. So values that are similar to desert or shrubland. And then it increased a little bit in the Oligocene, but didn't increase enough for us to interpret it as forest until the early to middle Miocene. We also looked at stable carbon and oxygen isotopes to reconstruct local climate, and that showed us that starting around the middle Eocene, we have very dry climates in this region, warm but quite dry climates, and climates don't get wet until around the early Miocene. So this led us to come up with a different reconstruction, namely that there was a shift here in the middle to late Eocene towards open, arid shrublands that had very few grasses, but had a lot of palms, because we see a lot of palms in the phytolith record. So this is a vegetation type that is completely non-analog with what we see today. We don't have any open arid shrublands dominated by palms, but it is consistent, or was consistent perhaps, with the idea that these were open habitats where there was a lot of volcanic ash blowing around because there was a lot of volcanism, and also perhaps a lot of dust and soil that these animals were eating. And so perhaps that was what triggered the evolution of Hypsodonti in Patagonia, South America. Okay, so in other words, when we summarize the information from these regions, we can conclude that one interpretation could be that Hypsodonti evolution is linked to aridity, not necessarily grasslands. Okay, so now I want to talk about C4 open habitat grasses when they diversified and spread. So let's first, and the case study is going to be East Africa. And let me first talk to you about what C4 photosynthesis is. So we call the type of photosynthesis that most photosynthetic organisms have C3 photosynthesis. And the C3 here is just because there is a molecule within photosynthesis that has 3 carbons. That's sort of the ancestral type of photosynthesis. C4 photosynthesis is a type of photosynthesis that is a solution to the problem of what's called photorespiration, which is that oxygen in high oxygen atmospheres, such as what we have today, oxygen interferes with photosynthesis. And so in the atmosphere that photosynthesis evolved in, we had very high CO2 levels, almost or no oxygen, no free oxygen. And so there we didn't have the problem of photorespiration, but now we do. And it also turns out that this type of photosynthesis, C4 photosynthesis, is advantageous where photorespiration is extra difficult, namely hot and arid climates and when CO2 is low. And also it turns out in summer rain climates. And this graph is showing some of this, that C4 grasses are favored high temperatures, low atmospheric CO2. C3 grasses are favored over here. And so because of this, C4 plants tend to dominate at low elevation and low latitudes. So all the orange here is C4 dominated grasslands and savannas. Okay, now if we look at the evolution of C4 grasses, so grasses with C4 photosynthesis, there are other plants that also use this type of photosynthesis. It happened several times within a particular clade, namely the clade of tropical grasses, so the pack mat grasses. And so C4 grasses are the pack mats that dominate in today's savannas and grasslands in the tropics. And this, if we look at our dated phylogeny, we think that this happened as early as the Eocene, so a very long time ago, that we starting to get evolutionary origins independent within the pack mats of C4 photosynthesis. Okay, now the great thing is that we can actually differentiate between C4 biomass and C3 biomass using stable carbon isotopes, because this process of photosynthesis is creating a different signal. So C4 plants tend to have much heavier stable carbon isotopic signal than C3 plants. Okay, so people have used this to reconstruct the sort of changes in C3 in the stable carbon isotope signal on different continents. So here, for example, in Southeast Asia, we see the age here on the x-axis, and then we see that there's a shift here towards higher or heavier carbon signal. So that is more C4 over here than here. So a shift here at around between 8 and 6 million years. If we look at Australia, the same thing. We see this shift from lighter to heavier carbon at somewhere after 3 million years. And so the stable carbon isotope record, or delta 13C, shows that C4 grasslands spread at low to mid latitudes between 10 and 3 million years, something like that. But the timing varies depending on the continent and region that we look at. So there's some variation. Phytoliths have also been useful in sort of documenting this change. So some work that we've done in the Central Great Plains and also in Montana, Idaho, shows that C4 grasses, a specific group called chloridoids, started to be present in the Great Plains at around 19 million years ago and became much more abundant after 10 million years ago. And this not just chloridoids, but other packmat grasses as well. And because we have chloridoids in this record from both the Central Great Plains and Montana, Idaho, we are suggesting that may show that there was a shift towards more arid climates, because chloridoids today are typically very arid adapted. So it could be driven by that potentially. And importantly, C4 grasslands in North America replaced C3 or Poaceae grasslands. So that's what I talked about before for the early to middle Miocene. Okay, so if we take these data together, it seems that, again, like there's a decoupling between taxonomic diversification of C4 grasses and ecological expansion, which happened quite late in the game. And also that there's a different in timing and vegetational context for the spread of C4 grasses depending on the continent. So I've added to this sort of view of the different regions when we think C4 grasslands came about in these different areas. It never happened in the Eastern Mediterranean. So now we're going to shift focus to East Africa. So the data that we had before we did the work that I'm going to talk about is that stable carbon isotopes were showing that C4 grasslands started spreading after 10 million years ago. And this was after we see hypsilon animals showing up in this area and diversifying. And this area is, of course, of specific interest to us because this is where our own lineage evolved. And the classical story was that humans came down from the trees and evolved many of our locomotory and sort of our big brains and our hands and so on because we're coming down from the trees and living in the savanna. So it's long been linked to the spread of grasslands, at least theoretically. It's been debated as well, as you might be familiar with. There are some pollen data that and some tooth wear data that points in the direction of grasses being important right around the time when we see hypsilon evolving, but it hasn't really been clear cut. So that's something that a former postdoc of mine, Alice Novello, wanted to look at. And so she was interested in investigating when open grass-dominated habitats appeared in Eastern Africa and what were the regional patterns of vegetation prior to the C4 grassland expansion as indicated by isotopic data. So to do that, she went and did field work in East Africa, and also we got samples from colleagues. This was a project looking at a couple of floras in Ethiopia from the late Oligocene and early Miocene. These figures are from Mush Valley, that's about 22 million years, so early Miocene. And there we had the opportunity to look at both macrofossils in the form of leaves and other reproductive structures, as well as sedimentology and geochemistry, and also, of course, phytoliths. So this was a very proxy project. And what we found there, both from all lines of evidence, pointed to a close canopy forest with forest grasses in one of the early diverging lineages. So definitely not grasslands at this point, or grassy vegetation. Then we've also worked some with a big collaboration to look at the evolution of hominoids called REACH. And so here are some of the people involved in that project. Alice and I worked most closely with Rahab Kinanjui of the National Museums of Kenya, who is a palynologist and also works on phytoliths. So the sites that we looked at for this project were early Miocene in Uganda and Kenya, shown here by these red areas, and they spanned in time from about 21 million years to about 16 and a half. And this was also a project with a lot of different proxies used, or lines of evidence: sedimentology, geochemistry, phytoliths, and also faunal data. Unfortunately, most of these sites didn't produce phytoliths, but Rusinga, Bukwa, and Moroto did. So I'll talk about those data. And so we looked at those sites. We also looked at the other sites. And even though those other sites didn't yield a lot of data, we did find some grass phytoliths. Most of those grass phytoliths that we found overall were from the pack-mat clades, so the tropical grasses, in which we have our C4 grasses. But we also found some Poaceae, so these are more cool temperate, and some bamboos or early diverging grasses, which are more forest grasses. Then we analyzed our samples by comparing them to modern soil samples that we had extracted phytoliths from. So Alice extracted phytoliths from a range of different habitats or different vegetation types in Africa, and analyzed the phytolith assemblages and characterized them, and then we compared our fossils to that. Both in terms of the potential C4, percent potential C4 grass phytoliths, but also looking at the assemblage composition overall. This is a very complicated graph, so I'm going to walk you through it. So here we have the maximum potential C4 grass phytoliths, and these are our modern vegetation types. So going from almost no potential C4 grass phytoliths in the rainforest, all the way to grasslands and bushlands and shrublands that have a lot of grass phytoliths, C4 grass phytoliths. And this is comparing our fossils to that. And as you can see, there's a range from very low potential C4 grass phytoliths to very high. This is showing our pie charts, also again indicating that some of these assemblages have a lot of grass in them, a lot of potential C4 grasses. We also then use ordination. So this is a non-metric multidimensional scaling analysis or ordination, where we can see that the rainforest phytolith assemblages are over here. This is the modern grasslands, and so all open arid habitats. This is another type of tropical forest, and these are some grassy areas in that forest. And and our fossils in red here are falling somewhere between these more open forests and the sort of grasslands, shrublands, etc. in this analysis. So what we concluded then is that our fossil sites are showing a range of habitats, but many of them are grass dominated, and that the dominant grass type we had were potential C4 pack-mat grasses. Okay, then we also, like I said, we had some geochemistry, namely stable carbon isotopes from both soil organic matter, plant waxes, so N-alkanes, and pedogenic carbonates. And I'm not an expert on this, but what you can see here is that the green color here is what the values that you associate with forest, and yellow colors are values you associate with grasslands. And you can see that there are some that are way over on the left and some that are on the right, a little bit depending on the proxy used, but we see variation. So we see overall abundant C4 biomass, but there's a range from C3 to C4 dominated. And in terms of habitats, we think we see everything from open to more open habitats to closed forest. Both, and this variation is both within and between sites, because each of these are different sites. We don't see a clear trend through time. So if we then put our samples here in colors in the context of everything else that's been done in Eastern Africa, or actually Africa overall here, through time, so on the y-axis, we see that our values are similar to values that we associate with this expansion of C4 grasslands at the end of the Miocene. And so we concluded then that in the early Miocene, down here, we have heterogeneous habitats with some C4 dominated grassy environments as early as 20 to 18 million years ago, something like that. So if we then put our results here in context, we see that we get some C4 grassy habitats early on in the record. And we think that it's possible that the volcanism and tectonism in East Africa associated with the rift may have favored C4 grasses, because that constituted disturbance, which is something that favors grasses in general. They're very weedy and disturbance tolerant. This also has implication for the evolution of locomotion in these apes, because it showed that these apes with similar morphology to some of the more upright apes might have evolved this semi-upright locomotion because they were climbing in trees, but then also being on the ground. So this changes a little bit the perspective on how this type of locomotion evolved, which is cool. The overall picture I want you to see is that the timing and nature of grassland ecosystem evolution varies depending on the continent and area. So we cannot generalize. We have to look at each region separately to understand it better. And also in general, and sort of the sort of what that means is that there's a complex pattern that also means that there are complex processes. So not necessarily the same thing that is driving grassland evolution and grassland assembly in these different regions. So in order to understand these processes, we have to look at each region separately and also compare them. So the work that we're doing now with my current grad student Ben Lloyd and colleagues in Australia is look at Australia, because it is a very interesting region. There we see perhaps the latest spread of C4 grasslands in the areas that are known, even though there's pollen evidence that open grass-dominated habitats probably came before the spread of C4 grasses. And so we think that we have something like that, but again, the timing is much later than everywhere else. So my grad student Ben and all these colleagues, we are looking at phytoliths and geochemistry and pollen in marine cores around Australia. And we also did field work in the outback, sampling different sites. And last year, we went back and also did some more sampling in museums. And so we're trying to cover the Cenozoic to some degree. Unfortunately, phytolith recovery is very poor in Australia, but we do have some important parts of the record covered. So this is to be continued. The other thing we're doing in the lab and have been doing for over 10 years now is work to refine how we can use phytoliths to reconstruct grass and grassland evolution. As I mentioned, there's a lot of ecological variation within grasses, and if we can get more specific, we can capture some of that variation and interpret it from the fossil record. And this is just showing that the different grass subfamilies, all the different colors here, have very different climatic responses. So to do that, we are studying the evolution, or we're studying the 3D morphology of grasses and trying to map that on a grass phylogeny so we can look at how it evolves. This involves my former postdoc Tim Gallagher, some grad students, and a whole army of undergraduates through the years. So we are hoping to create this way a detailed phylogenetic key for fossil phytoliths. This is just showing how we're doing that. It's harder than one might think. We're extracting phytoliths. We are staining them with fluorescent dye because phytoliths don't autofluoresce, unlike pollen and organic material. And then we're using a confocal microscope, which is a laser-based microscope, to image them. And this creates a stack of images that we can then stitch together to form a mesh that we can then align so that they're all oriented the same way and simplify. And then we use something called, oh yeah, I'll get to that. So this is just to say that we now have over 6,000 of these 3D grass phytolith meshes or models from 200 species from across the Poaceae. So this is showing the grass phylogeny and some of these models that we have. And so we're using them to study phytoliths' shape and size evolution within the grasses. And we're using something called geometric morphometrics. I don't think, for the sake of time, I'm not going to go through this, but it's basically using coordinates to capture shape. And that's something, so it's different from like measuring width and length and things like that. It's a lot more detail and a lot more precise. And then we're using various machine learning methods to create a model from the modern phytoliths and then to be able to place our fossil phytoliths. I'm not going to talk about all this, the detail. But we've been able to use these models, so we're able to place fossil phytoliths that we've also imaged using the same methods and place them in our model to figure out where they belong. And so we've been testing some of the previous work that we did using traditional methods for taxonomic placements. And that work has been able to refine what we can tell. So we've been able to reconstruct that we had woody and herbaceous bamboos in the Eocene of Nebraska, for example. And we've also most recently, this paper we just submitted, have been able to show that the poids here that we had we've been able to place them a little bit more precisely in the phylogeny and say that they definitely are poids that are adapted to cold climates, which is very interesting because they were coexisting with palms in the Great Plains, and that they were actually been there since the Eocene. Okay, I also wanted to say that Ben has my grad student has been working on using actually not just machine learning, but convoluted neural networks. So very AI methods, very advanced methods to try to get even better at sort of taxonomic distinctions within phytoliths so that we can get better at using them in the fossil record. I want to conclude by saying that I hope I've been able to show you that grassland ecosystems on different continents have unique, complex histories, and that we have to study them across continents to really understand their evolutionary history and how that shapes how they are behaving and functioning today and in the future as climate is changing. And with that, I want to say thank you for your attention. And now I'm going to stop sharing here if I can figure out. Here we go.
All right. Well, thank you very much, Carolyn. I kind of feel like my brain is full now.
Sorry.
Does anyone have questions?
Fast and Furious. Any question? Yeah.
I was wondering if anybody's doing this kind of work in Asia.
Yeah. So so I actually tried for as a postdoc in Sweden. I tried to sample or extract phytoliths from China, Tibet, Mongolia, and to no avail. Like the preservation is in the places I looked, was very bad, except in a lake, a middle Mi or early to middle Miocene lake in China called Shanwang. There we had excellent recovery, and so I did work there. Other people have also tried to do work there. Yeah, I reviewed a paper once that was had a phytolith record from central China or north central China, but I never saw it published later, which is very strange. I don't know how to interpret that, but where they did have recovery. So I think I'm hoping it's just a matter of time before someone finds the right sediment. Um, I don't know if it like one reason potentially for that poor preservation is that preservation tends to be best in volcanically active regions where there's a lot of sort of opal A circulating, or like a lot of, sorry, sort of easily dissolvable silica, such as in glassy ash and stuff like that. Also diatomites. Diatom preservation is, of course, the same as phytolith preservation. So if you have diatoms, you usually have phytoliths preserved as well. But yes, more work should be done in Asia, for sure.
Okay. Does anyone else have any questions?
I do. I can't figure out the hand raising thing on here. I used to know it. Anyway, this is Shari. I was wondering if you had uh oh good papers that someone could read about the human evolution and the grassland. That just captured my imagination. And I know nothing about grass phytoliths except for what you just said here. So with
Yes,
that in mind.
most people don't. But yes. I mean, I'm trying to think if there's something. Are you looking for like a paper in, a scientific journal, or more like a popular treatment, something a little bit less technical?
You
Uh,
know, either one. I do have an anthropology background, which involves human evolution,
oh, okay,
um,
perfect.
and a geoscience background. But sometimes those popular things just are really nice to read, and I probably share it with a couple of my friends who are really into that too.
Hmm. Yeah, so there is a paper from, or set of papers from 2009 by Tim White and his group in Science, where they actually were trying to make the case that grasses were not important for. They were looking at Ardipithecus, which you're familiar with. One of these hominids or hominoids. This is where I'm like I can never remember which is which, but they were looking at that, the evolution of that, and a sort of environmental reconstruction, and where the phytoliths were actually showing some grass, but he didn't like that, so he kind of buried that story in the supplementary material. But I'm I bet that story probably had one of these summaries. In the, like in science, you can sometimes have a perspective on papers, and so there might be something like that. And then let's see, one. Um, I'm not an anthropologist, so I'm less familiar with sort of popular treatments. There is um, there is, of course, the papers that we published. One of them is about the locomotion of Moropithecus, um, Morotopithecus. Is that what it's called? Oh, I can't even remember. Where it's more of a summary rather than a paper, and that might be a good sort of entry point. That's from 2023. So that might be a good entry point and have some references to some of these more broad, papers that are a little bit more digestible. So I'm happy to share those papers. But yeah, I'm probably, unfortunately, not the best person to recommend something more popular. But that said, if you Google, if you Google Maurodopithecus or Ardipithecus, I'm sure that there are lots of sort of news coverage, or some of these coverage in New Scientist and things like that that you could read. Yeah, sorry, not the best person.
Oh, that's great. I appreciate it. Thank you.
Okay.
All right. Sandy has a question.
Yeah, I have several questions, and they're all about phytoliths. I am really fascinated with those, and I got a chance, and I learned about these at the Burke Museum, and I had a chance to talk with Carolyn many years ago a little bit about those. And I have some of my questions. I have several, and they're all kind of similar. Um, do all plants have phytoliths?
Uh, they're all major groups of plants, land plants, have taxa that have species that produce phytoliths, but not all species produce phytoliths. So, for example, there are ferns that have phytoliths, but not all ferns make phytoliths. There are flowering plants that make lots of flowering plants that make phytoliths, but not all of them do. So it varies a bit, but all major groups of land plants make phytoliths. So it probably evolved very early on, the ability to make them.
And has anybody figured out what the purpose of them is?
No. I mean, yes and no. It was something that I worked on a few years ago to try to sort of test it, but what has been suggested are the most popular suggestions are herbivore defense or deterrence. So they wear down your teeth or mouth parts if you're an insect. And so, they're not, they shorten your lifespan if, you need your teeth to eat. Um, and so that some subsequent work that we have done and others have done indicates that this holds true for insects and maybe small mammals like rodents and stuff like that, but probably not for large herbivores, even though they do wear the teeth. So herbivore deterrence is one, support is another. So structural support, that's another suggestion. Still debated. And then also silica has a lot of other advantages. For example, protecting plants against metal toxicity, protecting against sort of tissue collapse during aridity or sort of drought spells. Also, some defense against pathogens. So some of that is because there's like silica, like sort of I can't speak, like bodies of like mineral, like solid silica in there, but also silica or silicon is bioactive. So it kind of the silicon itself is advantageous, but not necessarily because it forms a solid body, but because it's bioactive. Yeah. So there are many people, some people want to call it an essential nutrient, but it isn't technically considered an essential nutrient.
Okay.
So possibly many functions, but probably different functions in different plants. And also, yeah, that's probably a fair answer, even though it's not very satisfying.
Okay, the picture behind you, it looks as if those are phytoliths, those roundish things. And
Mm-hmm.
so maybe they're different shapes because of the way the plant cells
Mm-hmm.
grow and
Yes.
the shape of the cells. determines the shape of the phytolith.
Yes, in many cases, yes. I mean, most cases, I would say. Well, many cases. So in this case, this is probably from a grass. This is a fossil, and these are trichomes, so like hairs, and these are other epidermal cells. And so yes, in many cases, it's just straight up the shape of the interior of the cell or the cell walls or something like that. In other cases, in grasses and in palms and some other plants, the shape seems to be it's not yeah it seems to be not independent exactly of the shape of the cell, but it's more complex, morphologically complex than a plant cell. So it seems to have complexity of shape, even though functionally I don't think it serves a purpose, the complexity of the shape. And those are formed in specialized cells called idioblasts. And so I don't even know why they have that shape. That is very unclear, but they're not always exactly like a cell.
Okay.
Yeah.
Wow. It sure is fascinating, and it's wonderful that you have that tool, I guess you could say, to
Hmm.
use to study the soil and habitats.
Yes, I agree. I like it.
Thank you. Thank you.
You're welcome.
All right, does anyone else have any questions? If not, I'll stop the recording.